Walk down any supermarket aisle today, and the dairy case is no longer just dairy. It is a sprawling landscape of plant extracts, squeezed oats, and pulverized nuts, all competing for the same spot in your morning coffee and cereal bowls. The pervasive assumption is that because these beverages look like milk and pour like milk, they are nutritionally equivalent to milk. But human physiology and biochemistry tell a very different story. When we peel back the marketing labels and look directly at the molecular reality of what is inside these cartons, a complex picture emerges. Understanding the true nutritional profiles of plant-based milks versus traditional dairy requires us to look beyond the packaging and examine the hard science of the liquids themselves. We are going to conduct a scientific, evidence-based comparison to reveal exactly what fuels our bodies and what falls short. Let us first establish the biological baseline. For thousands of years, mammalian milk has served as a primary source of nutrition for human populations adapted to digest it. Traditional dairy milk is an evolutionary fluid specifically designed by nature to sustain rapid growth and metabolic function in a young animal. Because of this biological imperative, it is exceptionally dense in macronutrients. We define macronutrients as the primary building blocks of our diet, specifically proteins, fats, and carbohydrates, which provide energy and vital structural materials for the human body. A standard cup of whole dairy milk delivers roughly 8 grams of high-quality protein, primarily in the forms of casein and whey. These are complete proteins, meaning they contain all 9 essential amino acids in the precise ratios that our bodies cannot synthesize on their own. Dairy milk also contains a highly complex fat matrix. The lipids in cow's milk are enclosed in a milk-fat globule membrane, a unique biological structure composed of phospholipids and specific proteins. Emerging research indicates that this membrane plays a significant role in how our bodies process dairy fat, potentially explaining why the saturated fat in dairy does not behave exactly like isolated saturated fats in terms of cardiovascular impact. Furthermore, traditional milk contains naturally occurring sugars in the form of lactose. For individuals who possess the genetic adaptation of lactase persistence, this lactose provides a steady source of energy while actively enhancing the absorption of essential minerals in the small intestine. But milk is not just about the large building blocks. It is also a highly efficient delivery system for micronutrients. We define micronutrients as the essential vitamins and minerals required by organisms in varying, often minute quantities to orchestrate a wide range of physiological functions, from bone synthesis to cellular repair. Dairy milk naturally contains high levels of calcium, phosphorus, potassium, and naturally occurring B vitamins, including vitamin B12, which is absolutely crucial for nerve function and DNA synthesis. This brings us to a critical concept in nutrition science known as bioavailability. Bioavailability refers to the proportion of a nutrient that is actually absorbed from the digestive tract and utilized for normal body functions, rather than just passing unabsorbed through the system. The calcium in dairy milk is highly bioavailable because it is packaged within a specific protein structure called a casein micelle. This micelle, alongside lactose and naturally occurring vitamin D, actively facilitates the absorption of calcium through the intestinal wall. When we begin our comparison of plant-based milks, this concept of bioavailability becomes the ultimate dividing line between what is written on a nutrition facts panel and what actually enters your bloodstream. If we are looking for a true one-to-one nutritional replacement for dairy milk, the scientific consensus points heavily towards soy milk. Made by soaking, grinding, and boiling soybeans, soy milk has been consumed in parts of the world for centuries. Nutritionally, it is the only plant-based milk that consistently matches the protein density of traditional cow's milk, offering 7 to 8 grams of protein per cup. Furthermore, soy protein is a complete protein, which is a rarity in the plant kingdom. However, soy milk is often surrounded by a pervasive myth regarding phytoestrogens, specifically compounds called isoflavones. A common fear-based assumption is that these plant-based compounds disrupt human hormones or feminize the body. This myth stems from early, flawed studies conducted on rodents. Rodents metabolize isoflavones vastly differently than humans do. Decades of rigorous, peer-reviewed clinical studies on human populations show that phytoestrogens do not act like human estrogen in the body. They bind to different cellular receptors and often exert a very weak or even antiestrogenic effect. In fact, research indicates that moderate consumption of soy is perfectly safe and may even offer protective cardiovascular benefits. From a macronutrient perspective, soy milk stands as a robust, evidence-based alternative to dairy. But soy is no longer the most popular alternative on the market. That title currently belongs to almond milk. The meteoric rise of almond milk is a fascinating study in food marketing versus food science. Consumers often choose almond milk for its low caloric density, with unsweetened versions containing as few as 30 calories per cup. But those missing calories represent missing nutrition. Consider the biological reality of a whole almond. A raw almond is a nutrient-dense powerhouse, rich in monounsaturated fats, vitamin E, and dietary fiber. However, the commercial extraction process fundamentally alters this profile. When almonds are pulverized and heavily filtered with water, the vast majority of the almond's natural protein, healthy fats, and cellular structure are left behind in the discarded pulp. What remains in the carton is essentially a heavily diluted almond extract. A typical commercially produced almond milk contains merely 1 gram of protein per cup. To artificially elevate its nutritional value and mimic the profile of dairy, manufacturers engage in heavy fortification. They add synthetic vitamins and minerals, such as calcium carbonate or calcium phosphate, to match the numbers you see on a carton of cow's milk. But remember the critical concept of bioavailability. The synthetic calcium added to almond milk tends to settle at the bottom of the carton and is not encased in the highly absorbable protein matrix found in dairy. Studies show that while you might consume the same amount of calcium on paper, your body absorbs significantly less of it from fortified almond milk compared to dairy milk. Then we have the current darling of the coffee shop, oat milk. Oat milk is celebrated for its creamy texture and natural sweetness, which makes it behave very much like traditional dairy in culinary applications. But that appealing sweetness has a specific biochemical origin. To transform a solid, fibrous oat into a smooth liquid, manufacturers utilize specific enzymes, primarily amylases. These enzymes are introduced during the milling process to aggressively break down the long, complex chains of oat starches into shorter, simple sugars, predominantly maltose. This brings us to the concept of metabolism, which encompasses the complex chemical processes occurring within a living cell or organism that are necessary for the maintenance of life, including exactly how we break down food for energy. When you consume oat milk, your metabolism processes it quite differently than dairy or soy. Because the enzymatic process has effectively pre-digested the complex carbohydrates, oat milk provokes a rapid glycemic response. Maltose has a remarkably high glycemic index. A standard cup of oat milk can contain up to 15 grams of carbohydrates, and even completely unsweetened versions have a noticeable, rapid impact on blood sugar levels. While whole oats themselves are a phenomenal source of soluble fiber, specifically beta-glucan, much of this beneficial fiber is lost during the straining process of commercial oat milk production. Therefore, from a purely biochemical standpoint, oat milk is less of a structural protein source and more of a liquid carbohydrate. As we deepen our scientific comparison, we must address the structural reality of commercial plant milks. Without the natural emulsifying properties found in mammalian milk, plant-based alternatives naturally separate into water and sediment. To create the smooth, homogeneous texture consumers expect, food scientists rely on a variety of additives. These include emulsifiers, thickeners, and stabilizers like carrageenan, gar gum, locust bean gum, and gellan gum. While these additives are generally recognized as safe by regulatory bodies for general consumption, emerging research is beginning to question their long-term impact on the gut microbiome. We define the gut microbiome as the complex community of trillions of microorganisms living in our intestinal tract that actively influences digestion, immune function, nutrient synthesis, and even our neurological health. Some scientific literature suggests that a high, continuous intake of certain synthetic emulsifiers can disrupt the delicate mucosal lining of the intestine. This disruption can alter the composition of the gut microbiome, potentially promoting low-grade inflammation in susceptible individuals. Traditional unflavored dairy milk, by contrast, contains exactly one ingredient. When evaluating these nutritional profiles, it is crucial to read the entire ingredient list, not just the macronutrient panel. Another critical factor in this comparison is the presence of added sugars. Dairy milk contains naturally occurring lactose, a disaccharide that provides a subtle sweetness without the severe metabolic spikes associated with refined sugars. Many plant-based milks, in their original or flavored forms, contain significant amounts of added cane sugar or syrups to improve palatability and mask the earthy flavors of the base plants. Consuming liquid sugar is one of the most efficient ways to spike insulin levels and contribute to metabolic dysfunction over time. If you are choosing a plant-based milk under the guise of improving your health, selecting a strictly unsweetened variety is an essential, evidence-based choice. So, how do we synthesize this complex biochemical landscape? The truth is that there is no universal winner in the debate between plant-based milks and dairy. The optimal choice depends entirely on your specific biological context, your metabolic health, and your dietary goals. If your primary objective is maximizing highly bioavailable complete protein, natural calcium, and essential micronutrients without consuming multiple synthetic ingredients, traditional dairy milk remains an unparalleled biological package. If you are lactose intolerant, managing a true dairy allergy, or choosing to avoid animal products, soy milk stands as the only plant-based alternative that genuinely rivals the protein density and amino acid profile of cow's milk. If you are strictly monitoring your overall caloric intake and do not rely on your beverage as a primary source of protein or critical micronutrients, unsweetened almond milk offers a low-calorie base for your daily routine. And if you are an endurance athlete looking for easily digestible carbohydrates immediately before a strenuous workout, the enzymatically produced maltose in oat milk might actually serve a highly functional purpose in your specific metabolism. The pervasive myth we must dismantle today is the idea of inherent nutritional superiority simply because a product is marketed as plant-based. A beverage is not automatically healthier merely because it is derived from a plant, especially when that plant has been heavily processed, stripped of its natural fiber, enzymatically altered to increase sugar content, and artificially fortified with synthetic vitamins to mimic an animal product. True nutrition science demands that we look at the entire physical matrix of a food. We must consider how the nutrients are packaged, how they interact with our digestive enzymes, and how efficiently they cross the intestinal barrier into our systemic circulation. As we navigate the ever-expanding options in the modern food supply, scientific skepticism is a highly valuable tool. By understanding the fundamental differences in macronutrients, the critical importance of nutrient bioavailability, and the hidden biochemical realities of modern food processing, we can make choices grounded in biological reality rather than marketing rhetoric. Whether you reach for dairy, soy, almond, or oat, you now possess the scientific framework to understand exactly what you are pouring into your glass and exactly how it fuels the complex machinery of your body.